if we can settle down, we will get the last session of our workshop started. So for this session, we are going to just have a panel discussion because we really thought that it would be important to summarize everything we have learned over the last 2 days, 1.5 days, discuss broader questions we didn't get an opportunity to discuss due to time. And finally, I think, summarize the key action items for both FDA and for industry so that we can take our action items back and work on them take them back. So for this panel, we are welcoming back a lot of our panelists. So I'm going to start with, we have Dr. Aaditya Bhatt from Amneal Pharmaceuticals, Mr. Bryan DeBarr from Beatrice, Dr. Markham from FDA, Dr. Hiren Patel from FDA, Dr. Sam Raney from FDA, Dr. Wang from FDA, and also joining us from, on this panel, Dr. Heather Abdulgafar. Heather is currently a senior research investigator at Insight Corporation, and he has over 8 years of experience at Taro Pharmaceuticals working on various processes related to dermal product development, including analytical research, formulation, pre-formulation, and in vitro assessments prior to joining Insight. We also have Dr. Eleftheria Tsakalozou. Dr. Tsakalozou is a lead pharmacologist within the Division of Quantitative Methods and Modeling within Office of Generic Drugs, and her expertise includes physiologically based pharmacokinetic modeling and simulation approaches for topical and transdermal products, also development of quantitative modeling and simulation tools to support bioequivalence. So with that, we will start this panel with audience questions, if there are any, before we go around the table with all of our panelists to talk about what are their top 2 or 3 action items from what we have learned from the workshop. And also, as they are pointing out the action items, we would also like them to tell us what would be a good next step to take to resolve that concern or discussion point as a community. So like I said, we'll start with the audience questions. If anybody has a burning question, if they want to go to microphone or...
Yeah, the question I would like to ask here is the same thing which has been discussed in various occasions, that during the development, we find that by using various parameters, we are unable to get the decline in the flux profile. We tried different time points, dose, volume, and we find that the decline is not happening even up to say 96 or up to 120 hours. So as per the guidance, what is the option available is wiping procedures. So how do we, what acceptance criteria we can keep for wiping the procedures? Like, can we keep a mass balance that, okay, 1 wiping procedures of say that cotton ball of dry or wet, and then we check the mass balance of the at different sections, and can we keep a criteria for 80 to 120 for a mass balance? So can you please throw some light on such type of a situation we come to where we have to no option available but to do the wiping, and what actions and what procedures we can do? Yeah, thank you.
So it sounds like an experiment. I think there's a research project there for you. Maybe you can get a good grad student to work on that. So I think a lot of it is, this is something that's not well published, right? Sam, we don't see like a publication with wiping procedures and timing of wiping. I haven't read any papers about that.
I can comment on that.
Yeah, go ahead, Sam. No, I'm always up for good research projects. Thanks.
Okay. Definitely an area where more research would be helpful, but excuse me. think it's difficult to. So let's assume that you've done everything during method development that anybody can do. We're going to assume that in about 10 seconds, but I want to be a little bit cautious there that there is a lot to learn. I continue to learn things myself, and I've been doing this for a while. So I think it is important to first understand, it actually is critical, have you really done everything you can to optimize the method? And we've systematically laid out several reasonable considerations for method development through the exercises today that give you a kind of a sense of if everything we've talked about that are among your options in terms of optimizing that method, modifying the method, if you've truly done all of those things, then I think we move on to the next part. If you have not done all of those things, then I think the really important message is please don't underestimate, and I'm not saying you haven't, but please don't underestimate the importance of method development and really knowing what needs to be developed, because we do see a lot of submissions where it is apparent that there really wasn't, we would have spent more effort to develop the method and would have done specific things that we feel are highly likely would have dramatically improved the IVPD profiles. So I think just exercising caution that well, we've done everything we can. Now let's move on to other options. I want to really get the message across. Be cautious about that, because that could end up being in a situation where you then move in a direction and you then end up getting questions about having inadequately developed your method because, maybe there was some obvious scientific things that could have been done there. So I wanted to spend some time to really emphasize that part of it. Assuming then that everything reasonable and feasible has been done, then the question is, what's the purpose of going to a dose duration study or, the wipe-off study? Is the purpose exclusively to create a J-max in the profile that then has multiple time points that decline after that? If that's the only reason, I would recommend don't do it. Just say, look, you can see the method development report illustrates this is the behavior of this drug and this dosage form. And there's a lot of variations we've tried in the method development. This is the nature of the reference product and frankly of our test product. And as you can see, are able to show this to be a sensitive, discriminating IVPT method. This is just the nature of the product. And we're able to show that for this product that is Q1, Q2, Q3 matched, IVRT profiles are the same, and the permeation profiles through IVPT are also the same. It is characteristic of the reference product profile. And for our calculations, J-max is unfortunately the last time point, or somewhere near the last time point. I think that is a perfectly reasonable path forward. If, however, the problem is that you can't develop a discriminating method using those study procedures, I think and you need to be able to show sensitivity some other ways or something, then I would actually go to say, well, as part of further method development, I would then explore using a different dose duration method and see if this, in fact, is a methodology that allows me to develop a more sensitive and discriminating methodology. And assuming that that works, then that's your path forward.
I think one important point is that if you are really having trouble, FDA is willing to meet with you. I think if you have your data.
Absolutely.
And bring that, send your data in and request the development meeting with us so we can, but you better have tried everything and have your data in order and have good data to present to us and methods and method validation for us to look at and with specific questions about what you would like us to help answer. Thanks.
Yeah, I highly encourage that because I thought it was worthwhile to answer the question because I think the general considerations are of broad interest, I think, to the audience and to product developers, aside from your specific product. But there are going to be details about your particular approach to method development, the particular data of your product. And I think it's very helpful for you to have a pre-ANDA meeting with the agency to be able to have a shared understanding of what exactly your situation represents and what a recommended path forward might be for you.
Okay, we are going to move on to the second question, but thank you for covering wipe off early on.
So my question is a bit different. I don't think we've covered it. It was in the guideline. There's a lot of talk about randomization, and in some of the earlier lectures, we talked about the skin thickness and making sure it was even for both the reference and the test. And I'm assuming in the pilot as well, we'd make sure it was equal for the alternate, so that we've got similarities. So how do you deal with the challenges of randomization, but also making things equal?
Thanks for the question. So when you talk about randomization, and I believe you're talking in reference to the conduct of the study, right? So I think the first thing is our recommendation is to use AB in a sequential cell. And I think as Dr. Raney pointed out that, when you are measuring the skin thickness, what we typically expect is, order them in a rank from lowest skin thickness to go in the highest and put it in a sequential. And if you do AB, your treatment and skin thickness are randomized. Yes.
Yeah, I think that's exactly right. But also to clarify, that speaks to distributing error just systematically, in an equitable way across the experiment. So the skin thicknesses are relatively normalized for the A population and the B population. The randomization is a different concept. The randomization has more to do with the randomization of whether a donor gets the sequence AB or whether a donor gets the sequence BA. Okay, so that the dosing sequence is what's randomized.
Anna, there is somebody behind you who's waiting if you.
Oh, okay.
I'm so sorry.
Go ahead.
Sorry about that. Okay, yeah, thank you so much for taking the time out to do this. So my question is regarding sort of the role of harmonizing glassware standards going forward. So I noticed like in one of the technical videos that there was like a little bit of variation in like where the meniscus reference point is, or like there's discussions of variation in thermal conductivity of the glass and how that might like influence the equilibration of the skin temperature. And I noticed like one of the posters even had some like abnormality with doming that was maybe attributed to the glassware. So like going forward, it seems like there's been like a lot of progress I've discussed with some of you, maybe a little bit too much, sorry about that, that there's been some progress in sort of beginning to standardize these things. I guess like how much more would you say can be achieved for standardization by standardizing the glassware used in these receptors?
I think there's actually a lot that's already been done. I think we speak of this historically. A lot of cells were made per lab and we had to standardize based on what the glass blowing tolerance was. But a lot of these automated systems that you're seeing provide CFAs, and the glassware is well within a tolerance and easily reproducible, and you can purchase and insert in a set, and that's been a huge benefit. With respect to the meniscus, that's where we had a discussion previously with the dot on the cell. That is more of a reference point, but you can mitigate something like that by you controlling the amount input in the cell. You can choose a volume and fill each cell to roughly around that meniscus, but it's the same across cells, so then you don't have volume differences. But I do think there's been a lot of advancements and a lot of help from vendors to ensure tight tolerances on glassware, on caps, on stir bars, on all kinds of settings.
If I understood your question differently, incorrectly, forgive me. But if the question was, are we moving toward a single model of diffusion cell that everyone is going to use. Was that your question?
Not quite, sorry.
Okay, good then, because I don't think that's the direction. We want to encourage innovation to really optimize the tools that investigators have to be able to have different options when they need to have method development needs to develop great methods. Yeah.
Okay,
Thank
I can ask. So first of all, thank you so much. It's great. Everything is great. I'm sorry, I'm center co-director, but that's the one I wasn't involved nearly as much because we know Sam runs the show. Sam really knows ins and outs of everything. And then we have Audrey and Dana and Priyanka. Okay, I have a funny question as a patient experience. I already had this grant. I had this Center of Complex Generic grant, and on June 22nd, 2022, I look at the pictures, right? I move the hand, right, and boom, I got the frozen shoulder or something. And I remember that next day I had to fly to Sweden. So like in 2 days I had to fly to Sweden. So I immediately canceled some data meeting for a student. I go to emergency room. Really, I can't leave to save my soul. I cannot lift it. So they look at me, you know, put the patch right there I have the picture at emergency room, and I got in emergency room the patch lidocaine 5% patch from Octavius. Okay, I got home with Octavius patch. Okay, it's standing there. I'm getting the stuff, and then they fill out the prescription. I go to Plymouth Road CVS pharmacy. I fill it out, and this actually comes as I'll tell you what it was. It was lidocaine patch from Par. And as a patient, I tell you, it was not the same. I put one on, one stays, another slightly slides, slightly, just so slightly. I'm sure that in IVPT, IVRT would be the same. And I remember having actually telecon with Sam, and I thought, oh my God, I'm experiencing complex generic issue here. I got substitution from one to another. It feels very different. I mean, I truly remember in the patient experience. So how do you catch it? Because like my patient AI, whatever, I know it's not the same. I'm sure IVRT, IVPT probably the same. I do not know, maybe they were approved by this same method, but maybe hydration is different or something is different. Okay, that's the end of my question. I'm sorry.
So I'm going to take this one because it's a little bit outside the scope. For the IVPT, IVRT, we primarily focus on topical semi-solids. And I apologize that you had a bad experience with the adhesion of one of them.
Experience, good experience is good. I mean,
Cytokine goes through. But I can tell you that for most of the patches, we typically do actually PK studies and adhesion studies to support approval and not IVPT and IVRT studies. So we'll pick that one up.
so that is never, those patches never approved by in vitro studies alone.
They are approved based on PK studies.
Okay, that's why we have this project, I understand. Okay, good. Thank you for enlightening me. Sorry
There are
for
different
my...
guidances for transdermal systems.
Okay.
And I don't think we've had a CRCG meeting on transdermal systems yet. Have we?
We come back.
Okay.
It's never too late to educate you, Director. Thank you.
Sure. So we'll take one last question for the room, and then we will go around the panel. So please go ahead.
Thanks so much. How important is the actual raw quantitative flux in micrograms per centimeter squared per hour at each time point to you when you're looking at bioequivalence versus just the form of the graph, you know, making sure that it hits a good J-max and then declines? Does it matter if you're off by a power of 10 versus what you expect?
When you say versus what you expect, what we expect is that the test at reference product under the same study conditions of a sensitive and discriminating study behave the same. In other words, that there's no significant difference between the two. So there isn't kind of an external expectation. There's purely the expectation of comparability. Now, in terms of the individual points, if Jmax and AUC and AMT were matched, but there was something strikingly different about the shape of the two profiles, I think that would be a fundamental concern. We use those, and for the most part, for mechanistic reasons, those are kind of good anchors for characterizing kind of very particular attributes of the comparability of these two curves, the IVPD profiles. But we tend to look at the profiles as a whole, understanding that, as you've seen, even among replicates from the same donor for the same product, there can be some variation.
Sure, I just wonder about the specific, the source of the skin in the actual, the reference products, IVPT tests. I guess I have no way of actually knowing. Was it cadaveric skin? Was it cryopreserved? Did they use fresh tissue? Am I using the skin from a 90-year-old cadaver? Because my flux will probably be higher if the epidermis is significantly atrophic compared to the reference.
So the reference product is not approved based upon an IVPT study. So this is not like, taking an oral dosage form for which you have a reference plasma PK profile for the reference product. In those situations, that's why I kind of started off with, well, what is it that you kind of saying, what do we expect? What we expect is that in the IVPT study, where the test and reference products are compared under those conditions, which will be the first time that reference product has been tested under that specific set of conditions, and that forms the basis for a reference. That's the reference for that reference product using these test parameters. Now, under the same test parameters, does the generic product behave the same way? Within the context of the fact that we are already controlling the fact that the ingredients are the same, the composition is the same, the arrangement of matter is the same, the drug release rate is the same. All of that is already pre-gated before you even get to this stage. So it's not even that the IVPT study alone is providing the evidence for bioequivalence. It's supporting the demonstration of bioequivalence within a larger context where all the failure modes for BE, for bioequivalence, are systematically controlled.
So all within the realm of our developed assay, not whatever other assays have been done.
Correct, yeah.
Very good. Thank you.
Thank you. So in the interest of time, we are going to start going around the panel. I did check with Heather and he is okay with going first. I will say that we have about a little over 24 minutes left. So each of you get a maximum of 3 minutes. Thank you.
So there are a couple of things that, for example, I took away from here, from this conference, that I really learned, no, I wouldn't say learned, but enjoyed, which is the emphasis on the no alcohol in the receptor solution, because this question keep coming up when you have a low permeating API. And this emphasis was very useful to go back and revisit. Things that I would like more information about, I think, is if we have one outlier donor that does not permeate, what do we do then? So we have 20 donors that permeating and one of them is zero. Do we remove it? How do we deal with such a situation, such a condition? So that's, I think, could be interesting for everyone. And the use of the extended study duration, that was also useful to know, going to beyond the 100 hours. That's interesting, but I think that will make the study duration costs, as we saw, much more expensive. So overall, I think the main question that I have now is that outlier donor, and I hope you can help me with it.
Thank you, Heather. Think Rong, you want?
So I really appreciated these 2 days workshop, and actually from a review standpoint, I really appreciate the video session and Sam's explanation on this, step-by-step explanation, and it really provides a lot of information, to the reviewer. And I realized, especially in the when mounting the skin to the donor, and also dosing the product to the skin. Actually, there are a lot of teeny tiny, steps you really need to pay attention to. And this might be the origin of the variability. And in future review process, definitely we will pay close attention to the submission, how the applicant will provide, we would expect the applicant to provide detailed stepwise and well-organized submission. This will really critical for agency's assessment. And also, I noticed aberrant data has been brought up for many times in these 2 days. I just want to share some of my thoughts on aberrant data. I think there are several factors to consider when assessing aberrant data. Whether the number of the aberrant values in IVPT study, whether it's just an isolated observation or there are multiple aberrant data across multiple donors. So when there are multiple observations, are they really outliers, or it's just because the study was not well controlled, which leads to high variable data? And also we should look into the aberrant data, whether the aberrant data is from the test treatment or predominantly in the reference treatment. As you know, the statistical analysis for IVPT study is the scaled reference scaled approach, and SWR, the reference variability, actually plays a role in BE determination, and also whether the inclusion or exclusion of aberrant data would impact the BE outcome. So I think my recommendation would be, applicants should meticulously document each step for their IVPT study, conducting investigation on the observed aberrant values to see if there are any assignable causes. If so, the aberrant data can be removed because it's considered protocol deviation, and the applicant should provide their rationale in terms of how to handle the aberrant data. If applicant propose to include or exclude certain aberrant data, they should provide their rationale, their their efforts to investigate how the their investigation efforts, and also provide rationale on inclusion or exclusion of the data in the data analysis.
Thank you, Rong. I think one note for us is, at the moment, since the guidance says to include all data, if you are planning on proposing data exclusion in your protocol, it will be helpful to discuss those prior to the submission when practically possible. Okay, Bryan.
Yeah, I think what I've taken from this is the guidance provides a list of recommendations, and whether you run down the center of those or choose to be outside of those, it's on our responsibility as industry and individuals to justify even things in line with the guidance. And that's been a very difficult thing to do. I think it speaks volumes to a topic we touched on, which is the quality management system, and just how important it is to get that integrated into your lab early. We know there's challenges with doing development alongside regulated studies, but the sooner you can get your quality oversight on individual procedures such as dosing, such as sampling, to ensure that it's scalable, that doing it on small scale in development, and then when you try and tighten in on a 15 or 20 donor study, you can't adhere to your quality management system. I think that can lead to a lot of problems through the development and ultimate pilot and pivotal studies. So I think what I'd like to see from this is continue to work with vendors to work on getting every aspect of this under some type of reproducible control, working with the agency to see if they have identified areas that either need tighter or can loosen some controls, even in some regards. I think there'd be a lot of efforts that we could make to improve the quality management system, to look at data across labs, across systems, and work together to improve these.
Thanks, Ghosh.
So my key takeaways from this meeting was that the IVPT involves the need for a very methodical approach and having dedicated personnel who can work on this in a methodical fashion with regards to method validation, a careful processing of data, and recording everything that you do along the way. I think that hopefully this meeting has put that in that thought into everyone, and we have a lot of folks listening in virtually as well. And I can appreciate that we had a lot of deficiencies in this area in the applications prior to the guidance document. And when the guidance document came out in 2022, draft guidance came out in 2022, there were some improvements, but I think hopefully post this workshop, we've captured an audience of folks that are interested and dedicated in working in this arena of IVPT, that we're going to see improvements in even further improvements and not just the amount, but the types of deficiencies that come in. There'll always be deficiencies, but we're hoping to minimize them and move them from a major deficiency to minor deficiencies as much as possible, things that can be fixed. And I appreciate that the audience has paid rapt attention to what has been going on here. Thank you so much.
Thanks, Markham. Eleftheria?
Yes. Hello, everyone. I would like to invite you to switch for the next 3 minutes, to switch your perspective a little bit and look at IVPT through the computational modeling lens. And this is a true outlier because we haven't spoken about computational modeling at all during the last couple of days. But at the FDA, we are looking into computational mechanistic modeling. We see a lot of potential in this type of approaches and methodologies. And from attending this meeting the last couple of days, I feel that there is a lot of data, a lot of maturity in the thinking that can go around IVPT, that can go into developing computational models in silico methodologies that can describe the data that is generated within the scope of an IVPT study. Computational models are essentially mathematical models. The more mechanistic ones, the ones that we feel that are more promising and robust, are the ones that take into account formulation composition information that you already have for your product and the RLD, Q3 attributes that you already have for your product and the reference standard, and they make predictions, they generate predictions based on the design, the parameters of your IVPT study on how the API will be released from the formulation, your product, or the RLD, and permeate through the skin and be collected in the receptor solution. So these are very mechanistic, comprehensive, and sophisticated models. They require a lot of knowledge to be built, and also there is a complex process for validating these models so that we can make sure that they are credible and provide reliable predictions. But they can be very useful in our case for IVPT, on IVPT studies, for 2 reasons. One, they account for drug product specific information. So they are specific for the RLD versus the RS versus the test product. And they are really accounting for Q3 attributes, but also they describe key components of your IVPT study. So solubility of the API in the receptor solution, the thickness of the skin, the hydration level of the skin. It can be the pH or the composition of the receptor solution. What is the sampling scheme? frequency in the sampling? What are the sampling volume? Is it a full receptor replacement, or is it the sampling? So these models can really help you improve the design of your IVPT study, and test what if scenarios that, of course, to some extent will need to be validated, will need to be confirmed. But they also help you explore scenarios that you don't have to really run the actual study, or you can confirm with a small data set. So we we encourage you to consider these type of methodologies as you are developing your product in your drug development program, to look at what would be the design space for your generic drug product, for instance. How can you deviate in terms of a Q3 parameter compared to the RLD, and still, under risk-based assessment scenario, be considered bioequivalent, right? And also, how can you improve the design of your IVPT study to get the desirable permeation profile? I've learned a lot the last couple of days. I really admire the dedication in understanding the factors that go into developing an IVPT study. And this is critical for modeling and simulation as well. So it all falls into place eventually. Thank you very much.
Thanks, Eleftheria.
Okay.
Yeah, thank you. First of all, these 2 days seminar was really educative in terms of IVPT. So just like to briefly go over, like, couple of years back when the agency published the October 2022 guidance of IVPT, it channelized our approach towards the IVPT, especially the discriminatory ability and all. But over these 2 days, what I learned is that method development aspect for the IVPT speaks volumes. There are several aspects, not just limited to measuring the controls, IVPT controls over the study duration, but we need to deep dive additional several other aspects during the IVPT study. What if we encounter certain aberrant numbers? So we need to diligently observe, identify, not just during the validation study, but also during the development. Identify those critical aspects, not just let's say, hey, this one outlier has been observed, but we have to deep dive and identify what aspect could have triggered those aberrant numbers and identify them and see if there is a scope to improvise in terms of method development. And if this is just an isolated event, we can properly address them during our study so that this is an evolving topic, not just in terms of the education during the method development, but also for industry as perspective. So the more we deep dive and introspect, the better it would be to channelize further and arrive at a robust IVPT approach that would help the industry during the future. So I think, and also it's pretty education driven effort from the agency perspective also. So I thank the agency also to guide us during our study, during our course of years to the industry, so that it helped a lot. Thank you.
Thank you.
Okay, so I would start by thanking few individuals. So I think first of all, thank you so much. I think last 2 days, thank you for, being here and online and sharing your challenges, feedback, comments. It really helps us understand, what are the rate limiting steps, because we are on the other side and looking at the data, and now talking in last 2 days, I learned a lot from, many of you whom I talked. And during the last 15 plus month of, organizing and planning committee, I interacted with a lot of representatives from industry, academia, CRO, and it was a great learning experience personally for me. I would also like to thank CRCG members who were working effortlessly. They are always available. So thank you so much. I think we had an extensive discussion. I just want to, end with a strong note that while the regulatory decisions are made based on the validation and pivotal study, we heard repetitively from multiple speakers how critical is the method development, and we discussed a lot during the workshop. But I have also noted personally as an assessor in the ANDA submission, there are certain drug products which had some inherent issue and there are challenges. But when we see in an incoming application, when the method development extremely hard work or dedication behind the method development, which makes the validation and pivotal study very clean data. So I would highly encourage you to spend some time and invest your resource to have a good IVPT method develop, and that's going to be helpful for the validating and make the IVPT pivotal study adequate. And we hope for first cycle approval for all IVPT study down the road. Thank you.
We have 7 minutes. We
Oh.
have like 7 minutes.
I thought by the time we got here, I'd have like 30 seconds. So. Oh, thank you. We want you to succeed. I think it's really important that you understand that. We celebrate approvals internally. And we see how hard you're working. We want you to succeed. I want you to kind of really, really understand that, because in deficiencies, it's no fun. We don't send you a letter saying, here's a million things that you did a great job on, right? You get the other side of it. So I think it's important for me to express. We recognize how much you invest into even competing in the space of developing generic drugs. All of us at the agency have a very sincere commitment to, we're here because we really think it's very, very important to make generic drugs available to patients. And we're very proud of the products that have been approved over the last many years as being very, very high quality generic products. These are great products that my own family will be using. So where does this begin? I think my comments are really going to begin, or maybe end now at the end of this, where they began, which is with research. I think there are some action items for all of us. And for FDA, I think it comes back to research. Our regulatory standards are evidence-based regulatory standards. And as I talked about in my talk that kind of opened this workshop, those recommendations were based upon a lot of research and evidence. And I think there is continued research for us to do in terms of an action item coming out of this workshop. And I think we have research to complete in terms of the challenges being experienced, both for you and for us, in assessing the sensitivity of an IVPT method, for example. To be able to have not a qualitative, but a quantitative guide for that would be very helpful. Outliers, they're a very small proportion of data sets, but they occur in almost every data set. So they're just a nature of the scenario we were dealing with. Being able to develop the evidence that we have a shared understanding of the optimal way to deal with that. Ongoing research on adaptive study designs. I think the take-home message is we have a lot of work to do going forward to continue to do research in this area and to improve the recommendations that we can provide for you. And we do this for you. And we, I feel like we're responsible for doing this for you under the Good Do for Research program. There's also the recommendations and our guidance then that evolve from that I think we have room to clarify. So I think in terms of action items for FDA, not only is there room for research, but room for clarifying recommendations relating to when, for example, do you move to a wipe-off study, or we should just call it a wipe off, should be a wash off study, because sometimes it's a matter of actually washing, having kind of refluxing washes of the, let's say, the cream on the surface of the skin, that actually very gently and very effectively removes it. It's just a small wipe off after that. But when do you move to that kind of a study? In terms of the profile shape recommendations, I think there's been better, we can do a better job of communicating the acceptability of J-Max as the last time point. I think there's a lot of action items for FDA that we've been taking notes that we're going to try to do better on. And I echo the importance that the CRCG plays in providing a mechanism for us to be able to have these conversations, these scientific deliberations that kind of move this field forward. But there's not just action items for FDA. I think there's also opportunities for example, equipment manufacturers to now have, through a forum like this, a much more specific understanding of the procedures and the challenges and the assessment conclusions that are trying to be drawn. And we rely and really value the risks that you've taken to invest into your own research and design and development and marketing and commercialization in these spaces. We do not take it for granted. And we're extremely grateful for the innovation that you've put in already and that I think you can continue to put in. Sometimes, it's a small thing. I had some conversations with people relating to the height of the donor compartment, that chimney. This is really a vestige from an earlier diffusion cell design that ended up being very helpful, useful, necessary for IVRT studies, where you're dealing with large, volumes of the product. And it's helpful to have a good height on that because you use that as a parameter in method development. But with an IVPT study where you have very small doses, getting access to dose effectively, to have just a simple innovation of different adapters there for the top can be, one of the many innovations I think that can come. And then in terms of applicants, product developers, I think continuing to make training available to your personnel, to the people who are actually conducting these studies, you have to have a relatively sophisticated understanding of a lot of things in order to be able to pull off a good IVPT study. And we've made resources available even just through the CRCG at the 2021 workshop on IVRT and IVPT and at this workshop. Collectively, it's just a few days of training. And I would highly encourage that as a qualification criterion, just as the basic training before you have someone starting to work in this space, that they watch, they sit, give them a cup of tea or coffee and let them watch, and feed them too, but let them watch a few days of these videos and really use this as an accelerant for becoming very knowledgeable about what types of things to be considering. Because I think you've been working with otherwise very smart people who may just not have had as much exposure as we've all had the luxury to have today. And then I would end on saying the quality management system is exceptionally important, and the documentation in order for the FDA to be able to reconstruct what happened, the processes, the infrastructure within the laboratory, and the documentation during the study that reconstructs how things went forward, I think are some areas for improvement that I think will help us to, help you to succeed. So other than saying thank you very much to the numerous people that it took to pull this together, and maybe I'll leave that for some others to do as well. Thank you, and I've learned a lot from you, and I really appreciated everything that all of you put into this workshop. Thank you.
Thank you so much, everyone on the panel, not only for keeping on time, but also giving us a lot to do. So with that, we will wrap up this current panel. So thank you so much, everyone, again. And
we must admit that Sam has the best sound effects, right? Them. Who knew he can do that?
Okay,
just for that.
Before I introduce Rob, I did want to mention that after the closing session for our virtual audience, we will continue with the equipment demonstration for the FDA team that we were not able to accommodate yesterday. And for the non-FDA team, if you can stay in this room, we do have an activity planned for you. So if you are willing to stay back a little while for us, we would like to continue to get your input on guidance improvement. So please do stay back if you can. With that, it's my pleasure to introduce Dr. Rob Lionberger. Dr. Lionberger is the director of the Office of Research and Standards within the Office of Generic Drugs. He leads implementation of the generic drug user fee science and research commitments, including both internal and external research, provides recommendations for pre-submission advice on complex generics through pre-NDA meetings, product-specific guidances, control correspondence, and prior to joining the FDA 18 years ago, Rob received his undergraduate degree in chemical engineering from Stanford, PhD from Princeton, postdoctoral training in University of Melbourne, and he was a faculty, again, at Michigan prior to coming to the FDA. So please join me in welcoming Rob.
All right. Thank you, Priyanka. Thank you to all of the organizing committee and the CRG staff that put together this workshop. I think it was a fantastic opportunity to move the science underlying generic drugs forward. And, you know, what I really like about these workshops, and I think this workshop specifically gets to one of the things that we had when we were developing the ideas and the vision for the CRGC, is to really have super deep dives into challenging topics where we really bring together the experts of this from FDA, from the industry, from the companies that support the industry into one place to really put the fundamental challenges on the table. And I think that's what, you know, happens here. And I think that doesn't happen very many other places where you have that level of open engagement to really think about and air what's the challenges, what's difficult. You hear reviewers talk about, here's what I see when I review the application, here's what I'm trying to do, here's what didn't work well. You have perspectives from the industry, I'm trying to do these studies, here are the things I see. You know, in the example here that really struck me was, you always see what we call outliers, aberrant data. You see it all the time. If you see it in every study, not really an aberration, it's a part of the reality of this. And so we have to think about different, and we have to open our mind thinking about, this is not like a PK study where outliers, you have to keep them because if you don't, you're biasing things. There's something you have to deal with and think about. But really putting those ideas on the table to discuss, you can see the posters and data analysis that FDA has done on this issue. That's really what this workshop is about and how it really helps move things forward. And this is really, you know, the Center for Complex Generics has a wide remit. And this is just one of the areas where we're really trying to do this, to make it possible to have more efficient, more effective development of generic products. And that comes from several things. It comes from innovation, innovative approaches that allow abbreviated approaches. Remember, if you weren't doing an IVPT study, what would you be doing? You'd be doing a comparative clinical endpoint study on thousands of patients. And here, you know, we're very particular. Show me, you know, the dose response. Show me the sensitivity and selectivity. Do we ever ask you to show the sensitivity of a clinical endpoint study? Probably not, because it's not possible. You know, we do make you put a placebo arm in the study, and a lot of times that can fail. All right, so remember, you know, that's one of the reasons we're doing this, is that it's part of the how do you come up with much more efficient, scientifically based, and probably much more sensitive methods for bioequivalence and approving generic versions of products that people depend on all day. Like we have Markham here, a dermatologist. He can tell, you know, if you haven't heard already, the importance of access to competition for the generic dermatological products. He goes to the dermatology meetings. That's what people are talking about. My patients can't afford their medications if you don't have generic competition. So it's really important. And what we do here is fantastic, and it's super unique. There's nowhere else that this happens. And that's why I think this forum for the Center for Complex Generics is so important to moving forward competition. And, you know, and then we've seen that there's an executive order from the president on this, that, you know, that competition and drug pricing is a priority as well. And we think that this is well aligned with that activity. And so I always learn a lot from these workshops, right? You know, like the Center for Complex Generics, you know, as office director, deal with a lot of different areas. So it's very valuable also for me just to go through and really understand the depth of some of the challenges that are going on in each area. And, you know, when I think of this area, I'm like, why is this so complicated? And I say, why are we tied up with all of this validation for BE studies? And I think about, why is it different than PK studies? And the thing that I was thinking about is, if you're validating a bioanalytical method, which is a lot of work, and we also try to be very flexible, right? We don't tell you you have to use a certain bioanalytical method, right? We say validate your method, you can optimize it. That's sort of what we're trying to do here, right? If you complain our guidances aren't specific enough, it's because we're giving you flexibility to use very different approaches, different apparatuses to optimize that. But as a consequence of that is you're not going to get specific, precise guidance on exactly how to do the study. But I think the difference with the bioanalytical one is, right, for a bioanalytical method, I can validate against the molecule, right? It's separate from the product. I can also do my validation separate from the human variability, right? I don't have to, in almost every case, don't have to validate my bioanalytical method on incurred samples, right? I don't need the human variability in my method validation. But when we're looking at these products here, right, that's the challenge, right, is you can't separate those things. They're in there together. And maybe that's a perspective to thinking about how we make these methods work more effectively in the future. But that seems to be the challenge here, right? You really have to do a pilot study with that source of variability from your skin sources to show that your method is meeting the expectations that it's going to be able to distinguish products, that you're doing the study correctly. But hopefully in the future, maybe we get some ways that that can be done where we separate out those. Can we validate an IVPT method for a molecule and know that it works over a range of products? Can we do the validation parts in a way that saves the variability, that limits the amount of the variability that you introduce from the human samples? So again, lots of potential innovation going forward. The other thing I've learned from this area and thinking about it is in the GDUFA science program, we focused a lot on innovative methods, right? What's the alternative to the clinical endpoint study? All right, we have a new approaches. But really, it's the recognition that going beyond that innovation step, the next step is an implementation step, right? How do you get people to be able to do that so that you get high first cycle approval rates of generic applications that use this approach, right? That's the endpoint measure. And I think that's why the CRGC is so important, because that's not one group that's responsible for that, right? So it's really great to see a lot of our bioequivalence reviewers here, the people who review your studies, right? Because they have to understand how these work and what companies are doing, but also the applicants and the people doing the studies for them, focusing on the implementation. And I think that's an important part of the innovation life cycle leading toward access to affordable medications. And this is just one example, and this is, I think, something that's generalizable across other areas that the Center for Complex Generics holds. And I think one of our next workshops later this year will be on particle size analysis, where, again, it's a similar type thing. How do you do this effectively? How do we review this effectively and move toward a system where people can develop safe and effective generic products really efficiently, because everyone's aligned on what the right scientifically sound approaches are. And following up on that, the other thing that came out of this from my thinking is, and you've heard this several times from the panel, one of the things that you can do is you can meet with FDA through our GDUFA pre-ANDA meeting program. And I think that's something that maybe we've emphasized in the past a little bit more the innovation side of that, is come meet with us about doing something other than a clinical endpoint study. But you can also use this type of programs, and I think we're beginning to recognize the value in being more open to meetings on that area, where the meetings are not about, I'm going to come and tell you why I don't need to do a clinical endpoint study and I want to do the IVPT. I want to meet with you because I want to get this IVPT study right, and I'm running into challenges. The IVPT studies and the guidances are a little bit different than the PK studies that we see in many of our product-specific guidances. Almost always, we know, everybody knows what the PK of the brand product is because the brand companies had to do PK studies. There's no mystery about what the PK is. But very few brand companies did an IVPT study. No, until you start your development, or until our FDA lab maybe does something to support our product-specific guidances, no one knows what the IVPT performance of that approved product is. And I think that raises, more, we want to give you more flexibility in the guidance, but it also can lead to some of the challenges that we run into here. But I think you want to be really open to, because as FDA, we are engaging through the meeting process when you run into challenging implementation details for some of these complex generic products. And I think that's something that we certainly are more open to and we're beginning to recognize. So I encourage you to use that venue if you're running into some of these difficult challenges. That's probably the best way to interact with FDA around this. If you send in a control correspondence, you get a written response back. And I think just, because of the complexity of these issues, you've got to have that discussion and that back and forth and be prepared for that. And I think that's something we're really open to, and I hope that you will take advantage of that as we move forward in the generic program. But I appreciate all the effort that you and the audience make, both our FDA reviewers and our product developers and the people that provide the equipment and methods and to do these innovative approaches. That's part of the community that will lead to affordable medications. And I thank you all for your contributions to that. And with that, we'll continue in person, but thank you for all the people online who are listening to this as well.